that our lab has generated looking at the sweet side of tree resistance. So we usually think about defenses and tree, like nasty toxins that trees make in terms of defense. But I want to talk about the role of sugars and what they might mean for defense, and thinking about just resistance theories and how that all might fit in there. Okay, so I'm going to start here by telling you something you probably already know, which is that global warming and droughts and heat waves, episodic droughts and heat waves, have been creating these acute and chronic levels of elevated stress in our forests, and this has led to outbreaks of forest insects and pests and diseases, which has led to these massive tree mortality events across large forested regions of the globe. And so I show a lot of pictures like this because a lot of what I focus on are these western conifer forests, but it's not just happening out west, it's actually this global phenomenon. So this is this great map from Henrik Hartmann in 2018, where every dot on here actually represents a mortality event since 1970, and you can see that they occur on every single continent with the exception of Antarctica. So it is definitely this global phenomenon, a global issue, and something that we care about because there's lots of different consequences for ecosystem function. But even though this is a big issue, we still don't have a lot of information when it comes to how trees actually die from drought. So we don't know which species are most vulnerable. We don't know which individuals within populations might actually be most susceptible. And we don't have very good quantitative kind of thresholds of physiological parameters that can help us predict mortality under these future climate scenarios. So because of that, in the last few decades, we've seen this incredible surge of research just focused on drought-induced tree mortality and mechanisms by which trees die, right? And so we know that trees die from a combination of hydraulic failure, so cavitation or embolism in the xylem. We know that there is some role of carbohydrates and carbon limitation that occurs under drought. And then, of course, these biotic agents, which are the most complex piece of this puzzle. And so because of that, ecophysiologists that develop these models tend to kind of ignore that complexity, right? They're very idiosyncratic, they're very hard to model. And so a lot of our models, actually all of them, when it comes to forest mortality, tend to only include parameters that have to do with tree physiological responses to stress. And there's really no good model that includes insect responses or tree chemistry, which I think is kind of that link between stress physiology and how insects and pathogens respond to that. So this is a little crazy to me because we know that insects play this significant role in causing massive tree mortality events. And we also know that tree chemistry matters for insect choice. So the metabolome of a tree is really what's dictating whether or not an insect will approach a tree, whether they'll choose to attack that tree, and whether a tree can actually withstand or resist that attack. So what we're interested in my lab is kind of thinking about what the susceptible chemical phenotypes, and I put in quotes, look like, because once we identify kind of the metabolomic profile, if you will, it would be great to be able to link that to remote sensing in order to monitor these landscapes and better assess risk, right? Okay, so why hasn't anybody done this before? Why am I spouting and pontificating about it? Because chemistry is important, but it is also very complex, right? So trees produce hundreds of thousands of multifunctional metabolites. This is this beautiful image from my good friend Laura Richards at University of Nevada, Reno, where every line here is an NMR spectra from a different species within the genus Piper, right? So this is showing you intraspecific chemical diversity, where every peak is a different metabolite or compound, and the height of the peak is the magnitude or the concentration in a sense. And so you can just see this incredible diversity just within a species. So you can imagine between species and then in response to stress. And because we're scientists, we like to take lots of complex metabolites and put them into boxes so that we can talk about them more easily. So historically in plant insect literature, what we've tended to see is this division of metabolites into three major groups. So first there's primary metabolites, which are like carbohydrates and proteins and lipids that are directly involved in growth and reproduction, but also thought to have just nutritive value for an insect, right? And then we have what we've been calling secondary metabolites. Someone literally told me they thought secondary metabolites were garbage metabolites, and I almost fell out of my chair, because they are not. They're typically thought of as defense compounds, but they have many other roles, which I'm going to talk about. And then hormones, so these molecules that are just basically, signals and kind of regulating all of these organismal processes. But what we've learned is that by kind of dividing these into three separate categories doesn't really allow us to take a broad view of their multifunctionality, especially in these secondary metabolites, right? And so people have shown that not only do they play direct roles in growth and reproduction, but they can also serve as signaling molecules in and of themselves, in addition to defense and lots of other kind of stress-related responses. And so I just want to point this out because I say defense, but really these compounds can do lots of different things, and that's important to consider when we think about how trees are responding in terms of their secondary metabolism to stresses. Okay, so in our lab what we're interested in is how drought, I say and herbivory, but first I'll focus on drought, affects chemical defenses, and in order to do so you have to know something about the substrates from which they are produced, which are these non-structural carbohydrates. So non-structural carbohydrates are starch and soluble sugars, and they're important for both constitutive secondary metabolites as well as induced secondary metabolites. So constitutive is just there all the time, it's just kind of business as usual, where induced are the ones that are produced in response to a biotic challenge from an herbivore or pathogen. And so we care about them because both of these pools of secondary metabolites rely not only on recently fixed carbon from photosynthesis, but also carbon that's been stored away in terms of, basically starch that can be hydrolyzed into these sugars, right? And so what happens under drought stress is that this, input becomes constrained, and now we have a greater reliance on the stored NSC or carbohydrates that has to actually go into lots of different processes like maintenance, respiration, growth, maybe even more active storage, but also into these defensive compounds. And the way that they allocate this under stress has consequences for herbivores and pathogens that then have important feedbacks for tree survival. Okay, so the reason that I want to talk about NSC and active storage is because of this concept of the coordinated resource allocation hypothesis and the reserve safety margin, or RSM. So you can think of this as the NSC pool, right? So this is a paper I worked on. This was like a COVID project that we wrote up in 2021. And basically what it says is that this safety margin is actively put aside, right? And it can be a heritable trait. It can be this phenotypic kind of resiliency or plasticity that allows trees or plants in general just to be able to survive or be buffered against certain stresses, right? So we know that NSCs are used for defenses and for growth, but even though this is going on, there's really no trade-off because they tend to maintain this margin in order to buffer against future stresses. So that's the theory, right? And so trees are always responding to kind of supply and demand, and they're allocating resources in order to kind of balance those, the resources that are there with the sinks that are being generated by, let's say, an herbivore. But they also need to maintain this buffer. That's key. And so that's kind of what I wanted to think about when we were asking some of these research questions is, can NSC really tell us anything about how trees might be able to better resist biotic attack and survive? So what I want to talk about today is actually just two field projects that we've been doing for quite a while now, maybe for 4 years now. And what I want to do is start in the Southwest and think about bark beetles and ask these two questions. Do higher levels of NSC, or this reserve safety margin, does that translate to greater resiliency during drought and bark beetle attack? And then can drought-induced shifts in leaf level traits predict bark beetle attack? So They're bark beetles, so you're like, why do you care about leaves? But I will explain this. So we're going to first start here in the Southwest in pinyon pine, thinking about these questions. And then I want to talk about a different stress, herbivory, in aspen, and think about this intraspecific variation in NSC. So if it truly is heritable, and this is an actual active storage and trait, then can that determine the defense response and whether trees can survive these extensive defoliation events? So let's start here with these two questions, and we'll start with the first one. But before we dive in, I just want to kind of give you some background about bark beetle systems, and the one that I work in particular, because it's probably different than the ones you think about. So the system I work in is in pinyon pine, so it's the dominant conifer species in the four corner regions of the Southwest, so you can see in the screen here where pinyon is located. The work that we do is mostly kind of down here in south central New Mexico, but I've done lots of work in Colorado and Arizona as well. And so with pinyon pine, the primary biotic agent that causes mortality in the system is this cute little furry football helmeted creature here, which is the pinyon engraver. So this is a bark beetle. So what they do is they bore into the bark and they rely on the inner bark and the outer sapwood in order to grow and to reproduce. And so having nutrients available there, like carbs and protein, etc., and also maybe low defenses, is to their benefit because they're called secondary aggressives, which means that they can't really enter healthy trees. They actually only build up populations when they have lots of available host species across a landscape, which usually happens when these trees are injured or if they're experiencing severe stress like drought. So that is this little creature here, and that's what I'm going to focus on. And I just want to say one more thing about this, which is that these secondary aggressive beetles like Ips confusus, they're not mountain pine beetle. So they're not, we tend to make a lot of direct comparisons between what we know about mountain pine beetle and their ecology and say, oh, that must be how all beetles act. And this is ridiculous. And so what we're doing now is trying to understand these secondary aggressives ecology, and it's been very tricky, but very important, because really the impetus for this work was that when I was a grad student in the early 2000s, we were seeing this massive mortality happening in the Southwest in these pinyon juniper woodlands. So this is a pretty famous picture taken by Craig Allen in 2002, and you can see there's lots of trees in the red phase. These are all pinyon that are in the red phase, and you also see lots of green pinyon and some juniper there. And you think, oh, it's not so bad, some trees will die, some will survive. He went back and did repeat photography 2 years later, and you can see that almost all of the pinyon are dead. Every surviving tree is a juniper. And this has really important consequences for this ecosystem. Pinyon is an extremely slow-growing species, right? They're basically just glorified shrubs. That's how slowly they grow. And you can tell now that with all these dead pinyon, this ecosystem is going to be functioning very differently than it was before, and it may never recover given how long it takes them to grow. And so it would be great if we could predict which areas might be susceptible to drought and to bark beetles, and especially think about then what the ecological consequences might be when certain areas or if entire landscapes are affected. And so in order to do that, however, we need to know something about mechanisms, right? We need to identify drought-induced changes in sugars, in defenses, and also in how these bark beetles are responding to those shifts. And actually we have very little information how that actually happens in trees, and even rarer in mature trees. We know in saplings, because those are easy to work with, but no one wants to go out into the desert for some reason and do manipulative experiments. It's weird, right? Only me. No, I'm just kidding. It's not only me, but sometimes it feels that way. So this is actually our site. This is the Sabieta National Wildlife Refuge down in south central New Mexico, and this is a control plot. So these are just pinyons and junipers that are existing on the landscape and being exposed to ambient conditions, business as usual. And then what we did was, in 2020, we installed these plastic rainout shelters. And within one of the plots, we actually just diverted 45% of ambient precipitation away from 6 to 8 target trees in these plots. So they're about 40 by 40 meter plots. And so we installed this one in 2020, but we were lucky enough to also take advantage of the exact same manipulation, drought manipulation, that was installed in 2012 for a different experiment. What happened that year was bark beetles were coming through, and they were like, no, 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 we don't want these trees to die yet. So they sprayed them with pyrethrin, and by doing so, these trees actually avoided bark beetle attack and were allowed to acclimate to drought at this level for over a decade. So we call the one plot the 45 new, and the other one the 45 legacy, just to kind of keep them apart. And then that wasn't enough drought. We've done some other drought work, and we haven't killed them at 50%. So we were like, let's really just like torture these trees. So then we installed another plot where we diverted 75% of the precip away, and then of course we just love killing trees. So we were like, what about 90? Let's just see if they can survive 90. So we did, and really the 10% is just because we needed area around the tree to put in the structures. So we let all of this go, and then we let the bark beetles come in and tell us which chemotypes, which metabolic profiles they actually preferred. And so to our surprise, we've tried to kill trees in the past and we couldn't. To our surprise, we actually killed trees within 3 years. So like I said, the new structures were all installed in 2020. By 2021, we started to see trees die in the 90. And by 2022, we started to see a lot of mortality in our new structures. So we see trees dying in our new 45, in the 70, and in the 90. But we saw no trees die in these legacy plots where the trees could acclimate, and we saw no trees die in our control plots. What was up with this? So we wanted to know what was happening with the carbon budgets of these trees. Could that explain why some trees lived and some trees died? So what I want to show you is multiple lines of evidence provided by this incredible scientist, Dr. Drew Peltier, who was a postdoc on the project, who is now a professor at UNLV, and I want to show you some of the results that he generated to show that actually it takes a long time for trees to become carbon limited, and that may not be a good predictor of whether or not they die. So let's start at the beginning. So here on the left, what I'm showing you is just carbon flux, CO2 flux. This is vole respiration from trees that were in the 90s, so that's in red, so this kind of hard and fast drought. The control trees in blue, and then these legacy trees that were able to acclimate to kind of moderate drought conditions over the last decade. And what you can see is that relative to the control trees, we see that they now have a much lower vole respiration rate compared to the control, about 30% lower. But you can see these trees that have this really intense drought at the 90 are now increasing their respiration rates, right? So they've acclimated to actually lose less carbon through respiration, but they're also starting with less carbon. So when we look at how much carbon in terms of starch is actually in the sapwood that's being respired, we see that the starch levels in these acclimated trees are 75% lower than what they are in the controls or what they are in the 90. So 3 years of drought or 2 years of drought, this intense drought, doesn't really do anything in terms of overall starch content in the sapwood, but 10 years surely does reduce some things. So not only do they have less starch in their sapwood, and they're respiring less carbon from that sapwood, but the age of the carbon that they're using is very young. So what Drew was able to use is the carbon-14 signature in the atmosphere. So during the 50s and 60s, when we were setting off all these nuclear bombs and doing all this testing, we enriched the atmosphere with radiocarbon, with 14C. And the trees take up that 14C and they build carbon structures with it, right? So they stored away carbohydrates, they built biomass with it. And so what he does is he measures the respired carbon, and then he does some magic, and he's able to tell you the age of the carbon based on the exponential decay of the carbon-14 in the atmosphere. And so it's obviously much more complex than that, but what you can see here is that these trees that acclimated are actually using much younger carbon than the control trees or the 90% trees. And the reason for this is they've probably had to exhaust all of these older carbon sources just to survive under these sustained drought conditions. So they're really relying on new photosynthates in order to just maintain. The other kind of cool line of evidence that Drew found for this carbon limitation in these acclimated trees was the existence of these white rings. So trees have to, or usually form late wood, and that requires that the cells become lignified. And if you are carbon limited, however, you cannot really fully lignify the late wood formations, and so that leads to this white discoloration, basically. We see this a lot in trees that have been heavily defoliated, where they can't fix new carbon to sustain that carbon demand. But this is a drought-stressed tree, and what you can see, this is from November 2019, and in the 6 years prior, you can really see the carbon limitations start to show up, right, in these white rings. So, basically what we see is that trees under prolonged drought stress are carbon limited, but it really takes time. It took almost a decade in order to start to see the severe carbon limitation occur. So when we're doing these experiments, these drought experiments, and we just put them in this intense drought for 1 to 2 years, that's not really telling you much about how drought is really affecting trees, because drought is a cumulative metric, which makes it very hard to measure. So, it takes time. I've shown you some evidence for this, right? We saw in these acclimated trees reduced starch reserves in their sapwood. We see that they're using much younger carbon than control trees or these newly drought-stressed trees, and this white ring formation in recent years. So, what we see then, I can remind you, here are the acclimated trees, no mortality. So, they seem to be able to survive bark beetle attacks, right? But they have very low NSC. So this idea that having greater NSC can buffer you against biotic agents, well, if you're never attacked by them, we can't really test that. It's very, it's like my existential crisis these days. And so it's really led me to ask these questions. I mean, what does this mean for resiliency under future forest conditions, right? So I'm sure if a disturbance came through there, and those trees were to be attacked or to suffer in some, or to have to respond in some way, they would probably be toast. But I don't know that for certain. How long is this condition sustainable? How long can they stay like this, right? And then how are they avoiding bark beetle attack? That is wild to me. It is not that everything was attacked around our sites. It was really pretty focused on those plots where we put the new drought structures. So why weren't these trees that are basically living on the edge and have probably a very wimpy defense response, why aren't they being attacked? It could be that because they have such low carbohydrate reserves that beetles don't want to attack them because they can't live. It's like almost below this threshold, and there could be volatile signals or other ways that the beetles can perceive that. So it's not always about defense. There's other parts of the metabolome that is important that we need to consider. Okay, so we see that these trees died, right? Oh yeah.
Can I ask a question?
Yes, please.
What does it mean that a tree dies?
Oh, well, which hat should I wear? The physiologist hat?
I'm just kind of curious about it because it's really cool, right? To see these trees just keep going and going, and like, what is that switch that suddenly says,
I'm dead.
Nope, I'm done. Yeah.
I mean, that's a great question. That's really like, how do we measure like the point of, is it like this point of no return? Is it the point when they stop respiring? Is it like, meristem death? Is it, when they lose turgor beyond a certain level? So I mean, that's part of the problem with identifying when trees are actually dead. But I think for these trees, and this is something that we're actively researching, like I said, it's really the carbon economy, the water economy, and this defense economy. And I think when it comes to bark beetle attack, I think once a bark beetle enters and it's going to be successful, that's kind of like the biotic point of no return. The mechanism by which the tree dies, I think, is a combination of just exhausting carbohydrate reserves for a defense response, perhaps, like creating the sink that then takes carbohydrates away from being able to maintain turgor that is necessary to actually, so I think there's kind of, it's almost like it's like the Mannion spiral, like the domino effect. One thing kind of causes the other. Yeah.
So,
If I remember correctly, you mentioned that the legacy trees had been sprayed with
Oh,
something.
yes, yes.
So I feel like, would that also have an effect as to why they didn't get infested by the beetles?
That's a great question, and something we actually asked a lot about, because we're like, what is the legacy effect of this spraying? But it actually is pretty short term. Like, it breaks down fairly quickly, at least within one growing season. So what's been happening, so it kind of allowed them to avoid attack. They probably would have died if they weren't sprayed. But what it also shows us is that in a lot of these models, we're assuming that these trees in systems like this are just dying from drought, when in fact, if the beetles were not there, they would survive. Exactly. So it's funny that we're always like drought-induced mortality, when I think we should create another category that's like drought, but also beetle or pathogen-induced mortality, right? So I think in a lot of cases, drought, and even with pathogens, it's kind of counterintuitive. We think, oh, pathogens, they need water. Why would they attack this drought tree? But trees are full of water. And so if they're stressed by the drought, that actually makes it more conducive for them to survive. So drought kind of sets up this susceptible chemotype for them to then be successful. Yeah?
Yeah, kind of along the lines of the pathogens, are they, is there any evidence that they are evolving to deal with the changing environment and changing food sources and so forth?
Oh, yes. And that's the thing. So, I mean, I wish I had a soapbox I could just get on right here. So, yes, and that's so important to consider, because these different bark beetles actually vector different microbiota, right? So they don't all carry the same, we always think blue stain fungus, right? So they also vector a blue stain fungus like mountain pine beetle, but it is much different. It's actually a different species. Like, they're in Ophiostoma in the same genus, but very different species. And actually with these types of bark beetles that are kind of wimpier, a lot of the microbes that they vector are ones that are for nutrient acquisition. So they actually grow into ray parenchyma cells within the sapwood and tap into these lipid, protein, carbohydrate-rich cells, and then they extract nutrients back out into the beetle's galleries where their brood can actually feed on them. So it's not just pathogenic fungi. Some species do vector pathogenic fungi, but they also vector these kind of nutrient foraging fungi. There's also just kind of, fungi that are commensal. They don't really do it, you know what I mean? They're not affecting the beetle, but might be affecting the tree. We don't really know. There are also fungi that the beetle can vector that can detoxify. They can actually break down terpenes and other like phenolics, like stilbenes that are incredibly toxic, use them as carbon sources and make, that a more habitable environment for the beetle. So there's a lot going on, and so having system-specific information like this is really important, because, like I said, not every system is mountain pine beetle, which we always, those super aggressive beetles are very rare. Yeah, great question though. Yes, the microbiota is super important. Any other questions?
I'll ask some questions.
Yeah, please.
Ask some
No,
questions. It's great.
it's great.
This is a more basic question, I think, because that's how I think. But so how, how do these beetles sense whether a tree is drought stressed or not? And if you had, I realize that it's a widespread phenomenon.
Mm-hmm.
But if you had two trees, or I don't know if you could tell in your experiment, if you have a drought stressed tree and a non-stressed tree, will they attack the one and leave the other alone, and what's their sensing mechanism?
Well, right, and so that's the whole thing. It's like these trees are also drought stressed, but they've acclimated in a way that they are almost masked on the landscape. And so I wonder, so really what we think is that there's kind of these multimodal cues that they use. Some bark beetles tend to use visual cues. They prefer larger trees, because they need more phloem, but also volatiles that are being emitted from the tree that change under drought. And especially when other microbes are entering the tree, like in a stressed tree, these microbes release volatiles that also tell them, like, oh, this might be a good tree. We have not really identified in any system what those are. Some people think it's like ethylene or other, kind of just low molecular weight compounds. People have thought it was terpenes for a long time, but no one can really show it. So it's just, and they're also using kind of gustatory cues, and they use their pronotum to almost like use vibrations. They can vibrate against the tree and they can get information about anatomy and water, like sap flow almost. It's incredible. They can use, it's like, and then they can taste it and they can kind of say whether or not, they can almost predict, because it's all or nothing. For a bark beetle, once you make the decision to go in, you can't come out. You're not like, oh crap, there's too much resin here, like I'm backing up. It's like, you're done, and that's it. So you have to be really sure that that's the right tree, and so people have studied this for a long time, and it's lots of things, and we don't really have a clear sense of what becomes more or less important at different times during their, because it's also dependent on their density too. It's like if you have a lot of bark beetles around, maybe some of those things you cared about, if it was just a few of you, don't matter as much anymore, and you can overwhelm trees. That's like mountain pine beetle kind of mentality. So yeah, it's still an open question. Yeah. Great question. Yeah.
I have a related question. So I know like trees can talk to each other.
Oh, yeah.
So I know they would say like, hey, I'm having a drought or whatever, maybe the beetles here. Is it possible that the beetles are like tapping into that communication, or like, oh, they just told everyone they're stressed, so let's go?
Yeah, and actually yes, that is definitely possible. In pinyon, it's really interesting because they're the semi-arid species. They don't grow very close together because there's high competition for water, and so they're kind of spaced further apart. And so in terms of plant communication, we usually think they have to be much closer than these trees are, but we don't know for sure. They could definitely be talking. They also have these really incredible ectomycorrhizal networks below ground, which that is highly debated, but we do know that the presence of ectomycorrhizae in these systems can make the trees more resistant to drought stress for lots of reasons, but could also be a signaling thing in some cases.
I was also thinking about it with the death question, because I read Finding Mother Trees, so the
yeah,
only thing
that's a
I
great
know about
book.
trees is the mycelial network.
Yes!
So I was thinking, like, when they're about to die, don't they, like, send off...
Like a death rattle?
Yeah,
Yes,
they send
good.
off all of their nutrients. So would that also be a key to tell when they're dead, is when they're sending their stuff away?
It could be. In pinyon, it's hard because no one really wants to dig them. Like a lot of that signaling, or kind of like the exudates, obviously, are below ground, and no one wants to dig through these like rock beds, basically. But no, it's definitely a possibility, and even more than the trees kind of talking to each other. Another idea has been that when the bark beetle population density kind of reaches a certain level, that the pheromones, the aggregate pheromones that these bark beetles produce, can actually be sensed by the trees, and that they can almost prime themselves for attack. And be like, they can almost get their defenses and things that they're ready because they sense density, a higher density of bark beetles, because they've co-evolved with them for forever. So yeah, that's
Also,
something that we've been thinking about.
Another thought.
Yes, I love it.
Instead of like digging into the ground, couldn't you use like a similar technique with the radiation? If they're transporting carbon, you can sense the radiation through the ground.
Oh, the like carbon-14?
Yeah.
Yeah, that's something that Drew's been working on, actually just trying to be able to get a good read on the age of carbon in just the above ground defenses, like in the resin, because most of what's kind of getting sent out into the soils from the roots are like sugars, but also some of these secondary metabolites that can be used as signals, especially for mycorrhizae. And he's had some luck with that, but it's just really hard to collect and to measure root exudates. Yeah, I did a postdoc in soil ecology, and I swore I'd never go back. So I'm just
Sorry
like,
to traumatize
no, it's
you.
okay. I'm just reliving trauma. No, just kidding. No, but it's just so complicated, and so definitely a possibility, but really hard to measure. Yeah, and especially when they become drought stressed, lots of weird stuff starts happening, and it's just really hard to, it's hard enough to capture that above ground, so to try to capture it below ground is really tricky. Yeah. Great question, though. Anything else? Should I? Okay, well then I'll move on to just this other part of my, maybe this will be great. I could just talk about conifers and not aspen. Okay, so one thing, because it's my true love, one thing that we were wondering is, in these trees that died, could we measure shifts in phytochemicals in the canopy that could help us predict which trees were going to die, even if that shift or those chemicals have nothing to do with bark beetle success? So they're not directly maybe involved in mechanism, but they can tell us something about maybe a tree surpassing a certain threshold that makes them more vulnerable or susceptible, right? And so that leads us to the second question, which, this would be great to end on this note. Okay, so can drought-induced shifts in these leaf-level defenses or traits predict bark beetle attacks? So even if we don't know the mechanism, like your question, Ankur, it's like, can we at least improve our predictive capacity by just knowing something about shifts in chemistry that maybe portend these attacks? So when we usually think about defenses in conifers, we usually think about monoterpenes. And so the reason for this is because monoterpenes make up the bulk of the resin, right? So when you can imagine a bark beetle coming in, they get pitched out by all of this resin coming at them. And that's mostly monoterpenes, sesquiterpenes, and diterpene acids. But these monoterpenes are these 10-carbon cyclic compounds that we have a pretty good sense of how they actually play a role in mountain pine beetle ecology. And so they play crucial roles, but diverse roles, because at high concentrations, like limonene, for example, here's limonene. So limonene doesn't actually make up a lot of the, isn't large in terms of relative concentration, but when it does increase, it is really, really toxic to bark beetles. So when it increases, more toxic, but sometimes compounds are less toxic, like alpha-pinene, and actually when they are present at even just moderate levels, some of these bark beetles can actually use them to produce aggregate pheromones. So in some cases, it's very dose dependent. In some cases, they're toxic. In other cases, they can be used to the beetle's benefit. So even in Ips confusus, they usually produce their aggregate pheromones de novo in their midgut, but they can also take advantage of host chemistry with alpha-pinene, and they basically take it in, they oxidize it, and then they produce this positive cis-verbinol, which is an aggregate pheromone that they use to call in all of their friends. So that's probably, I would say, where most of the research has gone into thinking about resin chemistry and defenses and terpenes. But what I want to talk about for a little bit are phenolics. I feel like this has not gotten a lot of attention. Phenolics in general don't get a lot of attention in bark beetle ecology, but I think there can be some really interesting things for us to find, especially when it comes to being able to predict mortality, because hyperspectral remote sensing, the people that are good at it and know what they're doing, have convinced me that they can measure phenolics at the leaf level and from drones. And if that's the case, then that might be useful for management. So the reason that phenolics are of interest is that they're ubiquitous, they're produced by conifers, but every single plant on this earth also produces them. They're characterized by this aromatic benzene ring, and then they tend to have either carboxylic acid or these hydroxyl groups attached to them. And then these functional groups are actually modified and changed, and permutations that lead to this incredible diversity of these polyphenols or phenolics, and that is reflected in this incredible functional diversity that they have. So they play lots of different roles beyond structure. So one phenolic you probably think about a lot when you think of plants is lignin, right? Cell walls and structure and those types of things. But these phenolics can also do all this other stuff, right? So they can be directly involved in growth and reproduction. They actually play direct roles in cell division. This is probably the most important one that we think about with drought, is that they can actually serve as antioxidants. So under drought stress, these trees produce reactive oxygen species and free radicals. These phenolics can interact with them and strip that extra electron away, making them stable and protecting cellular integrity. So they're important during drought stress, but also we've shown that a lot of these phenolics can play direct roles in defense. So they have allelopathic properties or antimicrobial properties as well. Okay, so why do I care about them? Well, conifers produce phenolics in all of their organs. So in their leaves, and their stems, and their phloem, and parenchyma cells, and roots, etc. But the production across organs varies with drought. So this is a study that was done by Nadir Erbiljan at Alberta. And what he did was he measured phloem and foliage of lodgepole pine and the phloem from jack pine under drought conditions on the left and under ambient conditions on the right. And what you can see is that in both species, the drought reduced phenolic concentrations in the phloem, but increased phenolic concentrations in the foliage. So these kinds of relationships may become interesting only if the changes in the phloem are actually related to resistance against certain biotic agents. And so that does actually tend to be the case with Ips typographus. This is the European spruce beetle. So when we think about, mountain pine beetle in our western conifer forests, Ips typographus is kind of the analogy for the European system with Norway spruce. And what they see is that trees that survive the European spruce beetle attacks tend to have higher phloem phenolic content than trees that succumb. And so if there was a relationship between what's in the phloem and what's in the foliage, you might be able to make some links and be able to sense that remotely without having to do all this complex chemistry that we do. So our questions then were, can shifts in leaf phenolics predict bark beetle-induced tree mortality? And then could we use a general spectral model to predict those traits? So again, I just want to remind you who died and who didn't. So no death over here in controls and legacy, but we do have, death in the new 45, 70, and 90. And so we divided them into trees that died, but then we had the survivors, the legacy, but then the survived is actually just the control plot. So we separated them out because they're very different metabolically. But they show similar results. So here what I'm showing you is leaf phenolics, and you can see in the trees that died that they produced overall higher levels of leaf phenolics than those that survived, either in the legacy or in the control. And the idea might be that because they were very much drought stressed, that they were producing lots of phenolics in order to try to compensate for reactive oxygen species. What's interesting about this increase in phenolics in the trees that died is that that response was actually very much driven by decreasing water potential. So as the water potential becomes more and more negative, that means that the tree requires more and more tension, right? There's more and more tension on the transpiration stream, so they're more and more drought stressed. So we see that phenolics being produced in the leaves were responding to water potential. And we also see that in the trees that died, that the proportion of phenolics they were producing relative to trees that survived, so this is kind of the one line, this 100, this is the surviving trees, what they would be making relative to them. And you can see that at every single level of water potential, the trees that died were making more phenolics than the trees that survived. And you're like, this doesn't make any sense. They should be better protected. They're responding to the drought. They're protecting their cells, but they're just not doing it fast enough. So even though when they died, they had greater overall concentrations of phenolics, the rate at which they produced them was actually more sluggish than the trees that survived. So this is a graph of leaf phenolics as a function, again, of water potential for those that survived, died, and acclimated. And then what we did is we basically just created a water potential sensitivity value from the slopes of those lines, and you can see that the trees that died were less responsive to changes in water potential than those that survived. And so even though in the end they make a lot, it's almost like too much too late, that they probably were already experiencing levels of cellular damage from drought-induced free radicals that was probably irreversible by the time all these phenolics could be produced, for whatever reason. So then we've done some logistic regression and some other modeling, and what we see is that as phenolics increase, the probability that the tree died during the experiment tends to increase. And of course there's lots of overlap, so it's not the best predictor, but it's definitely one factor that we can use in our models to try to get a better sense of that threshold for when trees actually become more susceptible. So the next thing we did was we said, okay, well, they're pretty good predictors. Could we use a general spectral model to predict those traits? So even, let's say they were a great predictor, can we even measure them remotely in order to, go out and take a drone and see where maybe hot spots of susceptibility may be? And I didn't put these slides in here because Paul told me not to. And then, I'm just kidding. But what I'll say is that we did take leaf level hyperspectral reflectance data from all of our trees over 2 years, and then we took those reflectance data and we put them into a general spectral model one that was published in 2020 by Wang et al., a new phytologist. And this is a model that was created using neon data from lots of different tree species, mostly broadleaf, some conifers, but also taken from top of canopy, sunlit, healthy leaves. And they assured me that they were going to be able to predict the phenolics with high certainty. This model should be able to be used with any tree under any condition. And we put them in, and what we saw basically was that the model did okay. It could predict phenolics okay with an R-squared of 0.3 for the trees that survived, so the relatively healthy trees. But in the acclimated trees, there was no relationship, and in the trees that died, there was like a negative relationship, which is not what you want to see when you're doing modeling for traits. And so what that says to us is that these models are not robust when it comes to these kind of metabolic weirdos that are dying or acclimated in a weird way. And what that also says to me is that we need more studies that are measuring conifers perhaps, and not just broadleaves. They are very different. And also maybe we're not measuring the trait that we think we're measuring. And so even though we think we're measuring phenolics, it could be that we're measuring something that is more correlated with the phenolics at that time, but then under stress may not be there anymore, or may have changed in such a way that these models are not robust enough to actually capture that. So we're doing some work now to create a species-specific kind of spectral reflectance model with the traits that we've measured to see if we can improve those. But those are just some things that we've been thinking about. So I have 10 minutes. Let me just summarize this first, and then we can make a decision where we want to go. But basically, I just hope that if you learn anything from this, it's just that phenolics are multifunctional. Usually people think phenolics, plant defense, but that's not always the case. They do lots of other things that have real consequences for fitness beyond just defenses, like antioxidant properties. However, their role in bark beetle defense in North American systems is unknown. We actually know a lot about phenolics in the European systems with Norway spruce and spruce bark beetle. We know that actually those bark beetles vector a fungus that is highly pathogenic, and those stilbenes are really effective against those pathogenic fungi. We don't know really anything about North American systems in terms of phenolics or the fungi or the beetles or anything. What we do know in this system, at least, is that leaf phenolics tend to be indicative of its confusus-induced mortality risk. It's not the only predictor or the best predictor, but it's one of many that we're looking to build models with. And it would be great if we could potentially sense them remotely and find some sort of thresholds that may be indicative of susceptibility to bark beetles. And so the next logical step is to really look into these relationships between changes in leaf phenolics, which is telling you something about how a tree's stress physiology is changing with drought, and then key shifts in woody tissue chemistry. For instance, maybe phenolics, but also could be terpenes. Even though they're not mechanistically linked, doesn't mean they can't be correlated because of how stress responses are occurring in a tree. So looking for these relationships, and then more importantly, trying to link those changes to the bark beetle system itself. Are these even effective against bark beetles? How do the microbes respond? So doing real bioassay work and thinking about the system more holistically is going to really help us in our predictive capacity, because a lot of the things that we're seeing are incredibly species and system specific. So with that, I don't know, should we just talk about conifers, or should I tell you just a little bit about the aspens?
We can stop and see if there are some questions.
Yeah, I would love to take questions, yeah.
So you have a pretty good evidence that you can protect trees by getting them to adapt to these dry conditions. What could practitioners do, as a specific management strategy for these type of ecosystems to help give these trees the time they need to evolve?
So what people have kind of come up with is mostly pheromone baiting, or putting out pheromones that are repellent pheromones or things like that into areas where they know that the trees are high risk. That's been the most used strategy, and probably the one that the the feds have invested the most money in terms of like marketing and things like that. But, I mean, it's just not, you know, we can't change water regimes in the Southwest. Really, greater access to water is what they really need. But yeah, I would say probably pheromone baiting is the biggest one, that you can kind of be strategic if you know areas that are high risk. But again, that footprint is relatively small. So what people tend to want to do is to find kind of like the leading edge of where populations are growing and then put those out there. And so if some of this information could be useful for that, that could be one strategy. Yeah, it's a great question. Yeah?
Just out of curiosity, how do opinions respond if, you know, like a drought or, you know, a very long period of drought, oftentimes, you know, that's the case in an average or overall sense, but you have really wet years sometimes.
Yep.
You know, does that set them back enormously to have a wet year, or, you know, do they seem resilient and keep their drought posture?
Oh, I see what you're saying. Yeah, I don't really know. That's a great question. I think that's a general challenge when it comes to this type of work, is that we don't have lots of long-term acclimation studies, you know, because PhDs are only 3 years. And so so it's very rare to have kind of these long-term data sets. Like this was a total fluke, you know. It just, actually, thank God, sorry, I hope Will Pockman's not listening, but I'm glad that he never took this down. And so we got to really just kind of leverage something that was there just out of curiosity, and it really shows that acclimation is key. But to your question about whether a wet year would set them back, you know, they've definitely had like some wet years in this last decade, and at least from my N of 1 plot, I can tell you that it doesn't look like it's set these legacy trees back. You know, they've had some really intense monsoon years since then, and they seem to just be doing the same thing.
I mean, the California system's kind of poster child.
Yep.
You know, this decades-long drought or droughty, and suddenly a really water-full year.
Yeah, and I think, again, it's like the way that trees function. It's almost, you know, these are cumulative events, and there's some ecological memory, some legacy effects that we are just beyond what we have studied in the past. You know, we're looking at, and even in the study that we do, taking water potential measurements, that is an instantaneous measurement. I've seen a tree at -7 megapascals one day, and it rains, and the next day it's like -3.5, and so you're like, oh, this tree's doing great, and then it died like 2 weeks later. So, you know, it's like we take these instantaneous measurements, and then we try to say something about cumulative stress, you know, like in a tree, and it's kind of wacky. So we've been trying to actually even think about new metrics by which to think about drought, where it's like maybe if you do have lots of water potential data, you could say how long, you know, if you know where the net assimilation value is, so in Pinion it's around -2 megapascals, they close their stomata. Maybe if you know how long they spend below that net assimilation point, that might be a good metric to use to be like, oh, okay, this is cumulative, you know, days below here. Even though some days they're above, most of the days they might be below. But instantaneous water potential, we do instantaneous photosynthetic, you know, rates, and it's just, it's not cutting it anymore. And so that's where I think the power of merging, you know, kind of the dendro work with even the aging of the non-structural carbohydrates, like that gives you so much information about stress and how they've been using old versus new carbon just to survive over a decade. So, yeah, I think we're just trying to think of new ways to quantify drought and drought stress responses beyond just instantaneous clip on a leaf or snip a branch type approaches. Yeah.
So I'm wondering, like, is it possible that the correlation between your leaf defense and beetle attack is simply because you have more leaf defense the more stressed it is, and you have more mortality the more stressed it is?
Yeah. Yeah,
Okay.
I think it has to do actually with just, you know, there's kind of the elephant in the room, which is just genetics, right? And so we have no information about kind of just the level of genetic diversity that's across these landscapes. And I think that, that's something to consider, just how trees just respond because of how they're hardwired, but also in their ability to kind of be plastic. But then the phenolics, I think, are really just a measure of stress physiology. It's like if you're making more phenolics, you're responding to the drought. I think it does have a lot to do with antioxidant capacity. And it may have nothing to do, maybe it's linked to a weaker defense response, or maybe it's not, and that's what I mean. It's like chemistry could be really useful as a metric because the whole metabolism of a tree changes with drought stress. And so using those chemical shifts as metrics is really useful, even if they're not directly mechanistically involved in how bark beetles function. You know what I mean? It's like there's something about the change in the metabolome that's related to that change in the foliar chemistry that has made it susceptible. Even if we can't figure it out, it could be good for predicting, even if it's not good for understanding actual mechanism. Yeah. But I think that could be totally what's happening. Yes. Yeah.
This is a broader question, but the state of this field in terms of research, you showed these photos from 2004 that are really charismatic in what they signal about the landscape, and you also seem to indicate that this field has an underinvestment in research and there's not enough attention being given. So do you think it hasn't stayed on the top of people's minds since this particular mortality event, or that's not the kind of thing people focus on when they're deciding what to research?
That's a great question, and I think it's not that, there's lots of people interested in this and studying this. I think a lot of people, because of the damage that mountain pine beetle has done, have focused a lot on these super aggressive beetles like mountain pine beetle and Ips typographus. In terms of being understudied, it's really these secondary aggressives that now, because of climate change and these mega droughts that we're seeing, are now starting to emerge as major mortality agents. I told somebody when I started grad school and was studying pinyon and pinyon Ips that I told them, I said, oh yeah, we're seeing all these pinyon dying. And he thought I was lying. He was like, no. He's like, you must have the wrong tree. I'm like, no, I'm pretty sure it's pinyon. But because he had been studying pinyon forever and had never seen anything like it. So really this is a pretty recent phenomenon to see that level of mortality, and it's very much driven by climate change. And so, like so many other native species, I think we're going to start to see them. We usually think of them as just kind of background mortality in our models, but now we're going to really start seeing them emerge as major mortality factors, and I think that's where these systems have been understudied because no one could have foreseen this. It's like I said, when I told this professor that pinyon died, I thought was going to fall over. And so, again, that's why I wanted to emphasize, we know so much about mountain pine beetle, we don't even know everything. Do you know what I mean? And so these systems are even less studied, but are becoming really important, in the Southwest. And also, the thing is, if you're not living in the Southwest, very few people care about pinyon, which is sad to me. You know what I mean? It's I study it because I used to live there. I went to school in Colorado, but not a lot, if you ask people about pinyon, there's not a lot of people who know about it, and it's only in this one little niche Four Corners region. And so I think there's lots of reasons that some of these systems are understudied, but those are the few that I've kind of just seen in my very short time, working. I've worked in the system for maybe 10 or 15 years, but it's people care about it. They don't know. The other problem is that a lot of the work in pinyon or drought-induced tree mortality has really been on the ecofist side, really understanding. There was a debate for almost 15 years of whether trees died from carbon starvation or hydraulic failure, and it's both. It's they are together. And so we've kind of created these false dichotomies just in terms of the physiology, stress physiology, that has kind of led people down these rabbit holes. And the defense piece, and not even defense, just secondary metabolism, which is part of the bigger picture. That's why I showed that trichotomy. It's not a trichotomy. They're all working together. It's an entire syndrome. It's almost like you can't separate them. And so we've kind of pushed that aside, and I think at our peril, you know, it's like set us back to not consider the other part of metabolism and stress physiology research. And so that's kind of where, because I think it's always been a black box, like this is too complicated, but really it's not. It's like highly connected with what we already know, and so that's where I see our work kind of fitting in and making it less scary. Like you don't have to be a chemist to do this type of work. I'm certainly not a chemist, I'm like a pseudo-chemist, but I work with chemists and I have enough knowledge of the systems parts to put that together. I think lack of integration, kind of newly emerging kind of secondary pests becoming primary pests, and just this, you know, sense that things have to be separate, so like put us back a little bit. So I think people want to study it, and it's, I see a huge surge now, and I think that's great stuff. And with like the remote sensing, like that would be the holy grail, you know, to link all that. To really say beyond just like trait correlation, to be able to use that to say something about mechanism would be super powerful. That's like my dream, but you know, a lifetime of work. Yeah, but great, I mean, yeah, thanks for that question.
Really nice set of questions.
We're good.
I guess if there are other questions.
Okay, goodbye.
we can follow up with- Let's give her a nice round of applause.